It was a nice transition. All right, we'll go ahead and get started. Yeah. Oh, he had PowerPoints on it? Okay, I'll ask him. He might not want it because it might be something that might be privileged. Yeah, I gotta talk to him about it. I'll talk to him because I would like to have a question. Yeah. I told him to try to think about a type of question. All right, guys, let's go ahead and get started. We're running a little bit late today. Quiet. All right. So again, we have another faculty candidate today, and I have to meet with her at 11 o'clock. So office hours will start at 11:30 today. So, and they'll go to 2 because then I have to go to a seminar of the speaker. Okay, so that's the first thing. Just write these down. The other thing is we're almost done with. And if we get done a little bit early today, which I suspect we might, if people want to come like before, because I'm meeting with this speaker at 11, so if there's some time beforehand, I can meet with you guys as well if we need to. Just sort of a couple points. What we're probably going to be doing is we're going to do nucleic acids today, we're going to do polymers next class, and then we'll have 2 classes that can sort of be a review for the exam. Because there really isn't too much within nucleic acids, certainly not enough, in my opinion, to fill 2 classes. It's not like some of the other stuff that we've talked where I can just go on tangents about random stuff that's related to my research. But nucleic acids are certainly important. They're going to be a really important transition to, I think, biochemistry. I think that there's really fascinating things within here. So I'll try to sort of at least teach you guys a little bit about the structures of nucleic acids, tell you a little about how nucleic acids are sort of the building blocks of life and how they lead to synthesis of proteins. And what I'm probably going to do towards the end of the class is I found some really neat videos that talk about sort of transcription and translation, so the overall process. And so we'll sort of look at those videos because I think they're neat and that at least give you guys some sense of how actually amazing DNA kind of is in this sort of whole world processes. It's kind of really impressive stuff. And then again, next week we'll talk a little bit more about polymers, and then we're pretty much done. So, so with nucleic acids, we'll just start discussing the kind of structure of them. Okay. Yes. All right, so when we're talking about nucleic acids, the sort of first thing to discuss is the sort of base pairs. Okay, and there's really 2 classes of base pairs, or 2 groups of base pairs that we sort of talk about. We have the pyrimidine bases, so pyrimidine. So the pyrimidine base, sorry, not base pair, base. So this is pyrimidine right here. Okay, so this is pyrimidine. Okay, there's no actual pyrimidine base, but the 2 sort of nucleic acid bases that are sort of built off of this pyrimidine base pair are sort of what we're sort of focusing on here. So one of them is, there's actually 3 of them. So there's uracil. This is uracil. So So again, it's not important to memorize these. Maybe if you take a biochemistry course, in which case you can do it then, but I just want to sort of expose you guys to the structures. But occasionally, if you guys start to see amino acids and you see sort of a U drawn, it's referring to uracil, and this is the general structure of uracil. There will be a bond down there to the sort of sugars that we'll discuss in a moment. But this is one of the base pairs, uracil. Okay. The other one is it's thymine, okay, which is effectively uracil with a methyl group on it. So again, it's the exact same thing, but it's got a methyl group at that one position. We'll discuss that in a second. This is going to be a difference. It's one of the subtle base pair differences between DNA and RNA. Okay, and so if we see a T, what we're referring to then is thymine, and that's going to be the structure for thymine, right? And then the other one that's a little bit different will be cytosine, okay? And again, it's going to be very similar. Again, it's sort of based on the pyrimidine. It's like neon. Okay, so this is pyrimidine. The other one is cytosine. And again, it's very similar. You'll notice there's really very subtle differences between all of these, but they're all based upon that pyrimidine core there. Okay, so if you sort of look at this, right, sort of look within here, right? Right, this is effectively a tautomeric form, right? If you sort of push the electrons from here to here, yeah, Felix. That's pure. That is a pyrimidine. Pyrimidine is not a base, but these are all considered pyrimidine bases because they're based upon this structure. So when you hear the pyrimidine base, what they're referring to is the fact that that's pyrimidine, and all of these molecules kind of look like that, right? So again, just take those lone pairs and push them here. In this case, push them there, sort of draw the tautomeric form or the resonance form, and you'll see that built within each of these compounds is pyrimidine. So these are generally referred to as the sort of pyrimidine bases, right? So if you just sort of focus on that sort of central ring there, right? You basically have a six-membered ring that's aromatic that has nitrogens in this sort of 1,3 position. Okay, and this one here is called cytosine. And so occasionally what you might see is a C referring to a nucleic acid base, and that's going to be cytosine. So again, you notice that the differences are rather subtle. They each have sort of oxidation at this carbon here, where this has a proton. These are all sort of oxidized to the carbonyl. Then the difference between these two and this one is simply that you have an amine up there rather than oxygen. If you guys had to, why do you think we draw this as this tautomer? And this one is this tautomer. It's kind of, yeah. More has to do with the strength of the carbon double bond oxygen, right? We've seen time and time again that carbon-oxygen double bonds, you know, this is sort of this trend that we seem to see over and over and over again. I'm not even sure that I entirely understand the physical organic chemistry behind it, but it's one of those things you see a lot, right? It's the reason why the sort of enol-keto tautomer always favors the keto rather than enol, right? It's the same reason why an acetal, right, or or hydrate of a carbonyl is not going to be favored generally. It's usually going to be the carbonyl. Carbonyl bonds generally seem to want to form, and apparently carbon-nitrogen bonds. In this case, you have a carbon-nitrogen double bond here anyway, right? So instead of having it here, this is more closely resembles the aromatic structure, where this one doesn't, right? So maybe a question that I could ask you on the exam is, are these aromatic? I'll let you guys decide. Okay. The other one is purine bases. Okay. And so purine is so this is purine, okay? So in the same way, that's pyrimidine, this is purine, okay? And in the case of purine, there's a typo here. No, I didn't. Okay. Okay, so in the case of purine bases, we're going to see compounds that are sort of based on this scaffold, where we're going to see the bond in the same way that this nitrogen sort of is where the bonds are formed here. In this case, we're going to see the bonds formed here, and there's two that we're going to be referring to. One of them is adenine. Okay, so that's going to be adenine. Okay. And the other one, and if you see an A, that's going to be adenine. And the other one that we have is guanine. Now again, what I'm not going to ask you on an exam is to go ahead and draw guanine or pyrimidine or anything like that. But this is just sort of to recognize that these are the sort of major components within this. And right. Okay, so these are basically the base pairs. Okay, so this is sort of like analogous in many ways to when we talk about amino acids, right? We had these side chains on the amino acids, and we'll get into in a little bit the relationship between DNA and sort of amino acids. But these are going to be sort of the differing effects, right? So all the amino acids have that carboxylic acid and the amine, and then they have a different sort of branch point, right? And that's going to decide all the different ones. These are the major groups that really differentiate the different individual nucleic acids. Okay? All right, so with this in mind, this would be perfect if I have a board that I could throw up. If you guys have written this down, we'll sort of discuss this a little bit further. When we're talking about sort of nomenclature and extent, these would be considered the bases. In a sort of a natural system, the bases are going to be sort of bound to sugars. Okay, so when we actually see these groups, such as the ones that I've shown over here, what we're generally going to see is them bound to a sugar. Okay, so there's two different sugars that we'll see. So, for example, one sugar that they can be bound to is ribose. Okay. Okay, so this group here is ribose. Okay, and generally what you'll have, right, is over in this part is where you'll have your sort of nucleic acid base. So, say for example, adenine, for example, right, would have something like this. Right? So in this case, what you have is the sugar sort of bound to that base. And so at this point, now what we have is a compound that in total, this overall construct would be called a nucleoside. Okay? So the nucleoside is when you have your base attached to the sugar. So the sugar and the base. In this case, it's ribose. The other sugar that will sometimes be bound to will be deoxyribose. And so in a similar way, what you'll have is this. Different, nucleic acid over here. Right? And so in this case, we don't have an oxygen there, okay? And this is what's called deoxyribose. Okay? So as we'll see, right, once we start getting into sort of DNA versus RNA, RNA is often based upon ribose, as we'll see, and DNA is based upon deoxyribose. So the difference between the D and the R in these sort of names of DNA, RNA is really the different sugars, at least in terms of the name for them. Okay? So that is sort of building up now. Okay, now in terms of further nomenclature that you'll hear on occasion. Yeah, so there would be 2 hydrogens here. So there's no oxygen there. Yeah. So yeah, I just sort of left it blank. Okay, so further in terms of sort of nomenclature, the way that you would call these is based upon the sort of name. So if you have your base, right? Say for example your purines bases. And you have adenine. When it's bound to the sugar, right? If it's ribose, deoxyribose. Okay, so when adenine is bound to a ribonucleoside, when it's a ribonucleoside, so this is a nucleoside, and if it's got a ribo, then it's going to be a ribonucleoside. If it's a deoxy, then it's a deoxy. Right? So adenine becomes adenosine. Okay, when it's bound to the ribose, and when it's bound to deoxyribose, then the name simply becomes deoxyadenosine. So it's a pretty straightforward. Right? So for a molecule like adenine, if you have that adenine base pair that we saw before, the molecule itself would be called adenine. Once it's bound to a sugar, depending upon which sugar that is, if it's bound to a ribose, then it's going to be adenosine. If it's deoxyribose, it's going to be bound to it. It's going to be deoxyadenosine. Okay? Yeah, Charles.
We've learned so many reactions with regards to, molecules looking at what is your particular mechanism specifically that would shield these nucleic acids from reacting with any of the many reactions that we've learned?
Yeah, well, so the reactions that you guys have been learning throughout the semester have been largely not biological. right? We're talking about reactions on solvents and so forth with fairly strong things like, strong bases and stuff like that. Remember that the sort of compounds that we have, right, our blood is often, and sort of serum that's within us is often buffered solutions, right? Like, so you're generally somewhere around a pH of 7.5, and there may be reducing environments based upon, say, things inside the cell, or other environments based upon whereabouts you are within the body. But evolutionarily, these have been made to be stable. But yeah, you're right. I mean, if you take a molecule like this and you treat it with triple acid or sulfuric acid, things may be ripped apart, right? So just remember, there's a difference. We're moving into the sort of biological realm, so it's not the stuff that we're talking about necessarily having in a solution. But certainly chemistry is done on these to make these and so forth. So synthesizing these molecules actually has proved to be pretty lucrative to certain individuals. So, guanine. And we can talk about that in a little bit if I have time, which I'm sure I will. So again, you have guanine, and it's bound to a sort of ribose, right? Then what's it going to be called? It's not adenine, adenosine, it's not guanine, it's guanosine, right? And then, obviously, deoxy, guanosine, right? Pyrimidines, within the pyrimidines, right, you have cytosine, right, you have uracil, and you have thymine. Okay? Now, it turns out that in the case of pyrimidines, okay, what we have is 3. And what we'll see is that the one that's going to be constant through all of these is going to be cytosine. So we're going to see cytosine bases in both DNA and RNA. But in the case of uracil and thymine, what you're going to see is that uracil is predominantly in RNA, and thymine is predominantly in DNA. And so that's just one of these sort of artifacts that's taken place. I'm not quite sure why. Maybe there's some evolutionary reason why that is that you can learn about in your biochemistry course. In terms of the names, it gets a little bit strange here, though. These are cytidine. So it becomes like, I don't know why this is the case, but maybe it's the pyrimidine, it becomes cytidine, I don't know. Uridine, right? And then the sort of funny one, right, is that now you're sort of in the case of RNA, right, as I just said, thymine is in DNA, not RNA. So technically, thymine, ribo, or whatever you would call it, thymidine, right, really doesn't exist for ribo. So what they generally actually call it is 5-methyluridine. And again, that's just some probably archaic translation, again, probably because these molecules don't actually exist. So what it really is it's the uridine with a 5-methyl group, okay? So what you actually find is that in the case of thymine, thymidine is actually the deoxy one. So I don't know. I mean, this is kind of weird nomenclature. It's one of the reasons why I don't really expect you guys to memorize the nomenclature too much. And again, if you're sort of involved in the field, you'll get comfortable with it. But again, in these cases, you'll see deoxy, but then with thymidine, again, simply because thymidine is the natural one for DNA, it's just called thymidine. It's not really called deoxythymidine. Okay, so that is that. All right, so these are the general structures, okay? What we'll next discuss, I think there's probably a couple important things for you guys is sort of taking organic chemistry and translating to the next step. So here's the structures. It's important to sort of recognize these structures. The names might come up again, so now at least you sort of have a sense for why they are the way they are. I think one of the other important things as an organic chemist towards the end of the semester in terms of translating forward, because we're in organic chemistry, it's perhaps now worth it to think about the way that this sort of, you can have base pairs, right? So instead of a single base, oftentimes what happens in DNA, you guys are probably all well aware of the fact that DNA, right, exists as these helixes, sort of double helix, right, which really results because of the fact that what you'll have in these structures is groups of these molecules, okay, that are sort of built with one another, and then they're going to pair with another base pair on the other side. And so we'll discuss how that happens in terms of at least structurally. Okay. So the way that base pairs match up is there's really two sets. And this is going to be for DNA at least. So I'll ignore, we'll focus specifically on thymidine now. We'll sort of ignore uridine for now. We're going to focus specifically on DNA. And so within DNA, what you really see is two groups. A and T and C and G. These are going to be the two that are going to pair together and sort of show how this happens in a moment. Now, when you get these nucleic acids or nucleosides, I guess it would be sort of deoxyribonucleosides together, what ends up happening is you get a series of so I'm going to leave out anything there. So I'm just going to, there's two protons there, but I'm not going to show them. Right? This is deoxy. So the way that sort of nucleotides become oligonucleotides is that the sort of oxygens that are within them basically connect to one another through these phosphate bonds. Okay? So you can sort of read about how that's done and so forth. But basically this is the general backbone. In the same way that the amide backbone to like peptides is like the backbone to peptides, these sort of phosphate bonds that are really the linkages within, that sort of links all the sugar components together within nucleic acids. So a good sort of parallel, if you want to sort of think about it that way, is the sort of sugars represent the sort of amino acid backbone. So the carboxylic acid, the amine is kind of like this. And then the sort of side chains, right, that we see in amino acids within nucleic acids is generally the base pairs. Okay, so what you'll have molecule like this, right? So I'm just going to sort of give you guys examples of how a few base pairs can sort of match. Okay, and so this is going to be adenine and thymidine. Okay. And now I'm sort of showing in these nucleic acids basically the two purine bases. Okay, so what we have here is thymidine, right? Right. So the two, right? So we're going to have here is thymidine and cytosine. Okay. Did I get that right? Yeah, thymidine and cytosine. Okay. And what you notice about these two is in terms of a hydrogen bond network, this is what's going to bring these two together. What you notice is that it's going to be really three groups that are going to be important. Okay? It's these sort of groups here that you're going to notice effectively allow for the hydrogen bonding network to take place. Now, if you look at this oxygen, right, is that oxygen going to be a hydrogen bond acceptor or hydrogen bond donor? Right, it's going to be a hydrogen bond acceptor. It doesn't have any hydrogens. Right? Here's a nitrogen. Is it going to be an acceptor or donor? It's going to be an acceptor. But up here you have a hydrogen, right? So it's going to be a donor. Right? And it's the same thing up here. You have an acceptor, a donor, and an acceptor. And as we'll see, it's not necessarily that all of them will be that way, but at least in terms of looking at the parallels, you have these different basic acidic groups that are going to effectively form the hydrogen bond network that creates this sort of base pairs. Okay? On the other hand, when these sort of form a base pair, what we're going to see is, again, this is going to be the AT, so this is going to be thymidine and adenine, okay? And so for adenine, what we're going to see okay, so here's adenine, okay? This is thymidine with adenine, again, A with T. And what you see here, right, is that you have that really nice hydrogen bond network, and they line up really well. Where over here, you have a hydrogen bond donor, right, that's going to be able to interact with that hydrogen bond acceptor. And here you have a hydrogen bond acceptor that's going to interact with the hydrogen bond donor. And down here, we're not going to necessarily get any hydrogen bonding. So certainly a question that could come up on an exam, for example, would be that if I gave you this molecule and asked you to show how they undergo hydrogen bonding to one another, I'd ask you guys to line them up and show the hydrogen bonding. And that might help you be able to see how these pair up. In terms of cytosine, right, we're going to see that with guanine. It's always best if I draw from here. In this case, it differs because now we actually have that NH2 down here. Fairly poorly drawn. Right? But down here now what we have is we're going to have that extra hydrogen bond. So what we're going to have here now is we've got this extra appendage which has built within it an additional area for a new hydrogen bond. Okay? So in this case you have basically hydrogen bonding on the top, and you'll notice it's different, right? On the top one you have from the purine, right? You have a Lewis base on the sort of carbonyl here. Whereas down here, it's flip-flops. It's got the more acidic site, the sort of bronzed acid or the hydrogen bond donor, right? And it flip-flops over here on this nitrogen, right? We have a hydrogen bond donor, and down here we have a hydrogen bond acceptor. So it sort of flip-flops. It's got a sort of symmetry to it. And then as we sort of go down, now we have a hydrogen bond donor. Here we have a hydrogen bond acceptor. Right, on the other side, we have hydrogen bond acceptor, hydrogen bond donor. Here, we just got an oxygen hanging out. It's kind of like your pinky toe. It's probably a remnant of evolution that's not really necessary, or maybe it is, I don't know. But here, it now can sort of exist and have this additional hydrogen bond. Yes, Charles?
These cellular environments are all immersed in liquid. What prevents water from interfering with these hydrogen bonds?
What prevents hydrogen? So do you know, yeah, no, I mean, you asked a question that's pretty good. So, you know how Velcro works? What's that? No. Yeah, it's a whole bunch of tiny little, very weak hooks. On one hand, you got a tiny little hook. On the other hand, it's a very weak singular interaction. But the moment that you have a lot of those, they basically enhance one another. So this is something called avidity. It's this sort of idea that when you have, it's kind of the Velcro effect. It's like when you have one binding effect, and then nearby you have another binding effect, sort of a polymeric binding effect, where you have multiple small interactions can effectively make one big interaction. So you're right. You're not going to get this, right? And then you can read about primers and stuff. I'm not going to go into it. But even in biological settings, you need at least 4 of these to even start the process within priming, because it takes a few to get this hydrogen bond network starting. If you simply take adenine and thymine and put them together, they're not going to form a hydrogen bonding network, or if they do, it's going to be easily outcompeted by adventitious and extra water that's in there, right? It's the fact that you have multiple of them together that now allows multiple networks of that hydrogen bonding, and collectively, it's kind of like Velcro. It's this sort of avidity effect. Yes?
Why is that position of one? Why does it on the right? So, the end, let's say, on that time,
Okay.
and an opposition. And,
Here?
yeah.
Or is it?
Yeah.
I think those are just a different base pairs.
And one on the adenine.
Yeah, I mean, that's the beauty that makes these symmetric, right?
The bond is sort of right, is the other way. One up.
Oh. No, it should be fine. Should be fine. Oh, wait, maybe I missed something. Oh, yeah, I screwed up this. Is this what you're talking about?
No, that one. The one, the nitrogen bond is- No, the nitrogen bond is the one.
This?
Yeah.
And this?
No, look. Why are they're not?
They're not. It's the same, just spinning around. Right, see how the oxygen's here coming down? The oxygen's there going the reverse?
The nitrogen is facing down as well.
No, just take this and flip it over. We can talk later. Trust me. And it's am I missing something here?
No, you're right. Because for DNA, you have the box. And then for
Oh yeah. So I thought you were saying but they are still mirror images of one another, right? It's fine. So what I would say is you're always going to have 2 different strands. One's going to go, and that's what's sort of being said, is that you have the 5' to 3', right? Right. But anyways, within this whole system, right, you're going to have this group of hydrogen bond network that's going to effectively generate a fairly large network of hydrogen bonding in terms of 2 oligonucleotides, and they'll be coming together and they'll be forming this network. And it's this factor, and I'm not going to be able to draw necessarily this and create it into a double helix, but certainly you can look in your books and so forth and start to get some sort of pictures of them. Okay, so this is just the structure. The important point here is there's this hydrogen bond network. That's the most important thing that could be tested, right? So show the hydrogen bonds. Here's the molecules. 2 of them match up. The other 2 of them match up. I might not even tell you which ones match up with which ones, but I may ask you to show why. I may give you all of these groups and say which ones pair up and show the hydrogen bonding network that illustrates this or something like that. So that's what I want you guys to be able to do. I want you guys to recognize the hydrogen bond network, and I want you guys to be able to understand that A goes with T and C goes with G, right? And as you might imagine, once you get to RNA, right, instead of A going with T, A is going to instead go with U, because it's the exact same thing, it's just that there's a methyl group here in this one case. Okay? All right, so on to the sort of, yes? Wait, what?
Why does A pair with C essentially?
I just told you. What do you want it to pair with? Try to pair it with this one. You'll have a Lewis base and a Lewis base. There's not a hydrogen bond network.
So I'm thinking that if I want, for example, A and C, I can keep on an hydrogen bond, right?
A with C.
Yeah.
Sorry, A with C, right? But where? Right? Because that's what I was saying, is there's a symmetry. Here you have a Lewis base, right? You would have an oxygen with an oxygen. You're not going to generate a hydrogen bond between this and this. Here you have a hydrogen bond donor. Here you also have a hydrogen bond donor. So there's a symmetry to it. You could imagine an off shift, right? Is that what you're saying? Could it tweak and suddenly have a hydrogen bond lower? Yeah, and that's probably due to the secondary structure, right? So probably the way that these fold in terms of three-dimensional structures, this sort of lowest energy in terms of, say, for example, strain or sterics or so forth, probably doesn't allow for this one to sort of shift in that different way. Okay? All right. All right, so anyway, so moving on, this is DNA. Okay, and again, RNA is going to be the exact same thing, except that now we're not going to have, we're not going to have thymine, but instead, and we're going to have deoxyribose instead of, or sorry, we're going to have ribose instead of deoxyribose. Okay. All right, so in terms of moving forward with biochem, and again, this is certainly starting to get a little bit out of my wheelhouse, but I'll try to sort of put it into perspective, at least in terms of my general understanding, and then I'm going to show you guys a few videos that I thought were pretty cool that sort of illustrate this, okay? So RNA, so DNA is sort of like the template, right? DNA, you have your DNA sticks around, it's not really degraded, at least in your system. You don't have enzymes that degrade DNA, for example. You certainly have processes that replicate the DNA in your system and make more copies of that DNA. But DNA is kind of like the center, at least for us. It's a little bit different for viruses, but for humans, at least, the DNA is the sort of standard. That's why oftentimes when you guys are talking about it's in your DNA, right? They're not saying it's in your RNA. Because the DNA is really the crux of where everything starts. But sort of how the origins of everything take place is you get this sort of system where DNA generates RNA. Okay, so RNA is the template. Sorry, DNA is the template. DNA, as we'll see, makes RNA. RNA is kind of like the workhorse. The RNA effectively then makes you, right? It makes the proteins and everything else that effectively makes you are who you are. So your DNA code that you have in your body, the reason why we all look a little bit different, for example, could be because when it goes through this process, there's a specific number of proteins that are in fact made that might be different for you than somebody else, and sort of so forth, okay? So RNA, right, can effectively, as we sort of already stated, it's going to be ribonucleosides rather than, so from a structural standpoint, there's just those two subtle differences. And you can sort of subdivide RNA. That's sort of one of the confusing things is that there's a few different types of RNA that we'll often discuss. So, so there's a few that we can sort of ribosomal. Okay, so ribosomal RNA. Okay, this is the RNA that's responsible. Okay, we have transfer RNA. Okay, transfer RNA is basically it's the sort of we'll see this in a video. So the transfer RNA, we'll start talking about codons in a minute, but what we'll see is that, again, the RNA serves as the template, it's basically leads to the protein synthesis, and as we'll see, there's a series of codes within the RNA which will basically say what the protein sequence is. So there'll be a code, there'll be a sequence of that RNA sequence, and that RNA sequence will effectively give a sequence of amino acids which will make a specific protein. The sort of transfer RNA are effectively groups of nucleic acids that help this process, and they're generally bound to an amino acid. So they effectively carry within them a specific amino acid, and this sort of helps this process. The video will sort of, I'll show this and I'll slow down in parts to explain this a little bit more. And then what you have is the messenger RNA. Okay, and this is what's going to set the stage. Okay. So we're not really going to focus too much upon the ribosomal RNA. We're going to focus more on these two to sort of simplify the process. But effectively what happens, the way that, and we'll show videos about this in a second, effectively what happens, right, is that it's going to be way more complicated than this, and you'll see it in the video. There's all these other proteins that come in. But simply in terms of a sort of getting a general feel, the first thing that happens is the sort of messenger RNA copies info from DNA, okay, and carries it to the ribosome. The ribosome is this crazy machinery. It's basically where all the proteins are made. The ribosome is, again, we'll see a sort of video of it. It's pretty nuts, like how impressive the ribosome is. So again, the sort of first step that's going to take place is that you're going to have your strand of DNA, and we'll see a video of this in a second. And what will happen is through an enzymatic process, the system setup, this is what's called transcription. This sort of DNA will serve as a template, and you'll generate this messenger RNA, okay? The messenger RNA will then go to the ribosome, okay? Once the messenger RNA is in the system, what you end up having there is these two different RNAs, and these are going to effectively help the overall process. So the sort of ribosomal RNA work with the mRNA, and again, you can learn more about the details behind these different proteins in biochemistry to catalyze new protein Okay, so again, my understanding of the ribosomal RNA is really not, I don't fully understand how this one works per se within that bigger field, but certainly the mRNA is the sort of first step. The ribosomal RNA somehow is involved in this sort of process, but the other one that I think should make sense to you guys is the sort of transfer RNA, which is the tRNA, and as we sort of just said, these are the pieces Okay, so again, I'm going to sort of show a video that I found, and I'll post a link to this website online so you guys can view the video again, because I think this is a fantastic website. I think you can talk about all this stuff, but I think until you actually see it happening, it's just sort of like, okay, great, so what? It sounds cool, but a second. I just want to turn up the volume because they're going to talk. Okay, so this is going to be a video on transcription. Okay, so this is going to be the process whereby effectively we're going to start generating that messenger RNA. So this is going to be that first step. It's going to here what you're going to see, and you can sort of make out the coils a little bit here. And again, I'll post the link, and you can watch it if it's sort of difficult to see. But what you have here is the sort of helical coils, right? And up here is going to be where the sort of enzyme is going to start doing some things. Let me just turn up the volume.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins.
Now this is
transcription factors assemble and
just so that we're all. This is computationally. This isn't like okay, but it's kind of cool. It's real time.
Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place.
Okay. So basically what's happening here is you're forming a network of enzymes. So these are enzymes that are basically forming a network around the DNA, okay, and they're effectively forming this machinery. And they're saying the polymerase, right? So we were talking about ACEs, right? There's different ACEs for enzymes. A polymerase is something that takes something and makes a polymer. The polymer that they're referring to here is going to be the messenger RNA, okay?
It with the help of other factors between the strands of the DNA double helix.
The cofactors are just other proteins involved.
The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism.
So you see what's happening there? It's going down the whole base pair system, right? And as it's doing it, you got this little tail that's coming out, and that's actually all the RNA being generated as it's sort of reading, as it's sort of reading the tape, if you will.
The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied.
Okay, so that's important. A lot of the pairs that we have here within the DNA, right, it's double helix. What's happening here is it's only taking one side of that helix, and what you're doing is you're matching up, again, the sort of RNA, right? The DNA and RNA sides, again, you can have that sort of match within the RNA and DNA side of things in the same base pair region. But it's that sort of match that's going to allow the sort of generation of this, but it only wants the one half. So if you sort of look in this image here, it's kind of neat how they sort of show this. And you can sort of see what's happening here is the DNA is sort of being split, right? So you've got the helix, but then within there you have this sort of split, and what's happening is in part of it it's able to read the one side, and then what's happening is you get these ribonucleosides that are effectively getting in there, matching up, and are sort of continuing this overall process. And again, all these other things that you see here are those ribonucleases that are sort of free and that are in solution, and that keep
only one of
getting
the
in there.
strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined.
I don't know. You think, yeah, the enzyme is almost certainly moving the DNA and not the other way around. But it could be the two of them in concert in a way as well. I don't know. These are the type of things that if you're really interested in this kind of stuff, this is like the heart of biochemistry. So if you're really fascinated by this stuff, biochemistry, we'll go into a lot more depth about this. Okay, so this is the sort of, I think from an organic chemistry standpoint, you can go into a lot more detail on a lot of these other things. But this is the first part that I wanted to show you guys a video of. So this is kind of the This is what's called transcription, okay? And if we have time, I'll talk to you guys about maybe drugs and reverse transcriptase, which is what viruses use, which effectively do the opposite thing. And that's how they effectively make the DNA, which is then injected into other host cells and sort of spread, at least in terms of, okay. Now, I guess I'll just show the video for this. I thought this was a kind of nice video. There's a codon system in place as well, so I'll just show this video.
Once the DNA double helix had been discovered, the next big challenge was to work out how the 4 letters of DNA could code for each of the 20 amino acids.
These are all different amino acids.
The first question was, how many DNA letters coded for each amino acid? If it was 1 DNA letter for 1 amino acid, then you could only code for a maximum of 4 amino acids. 2 letters in every possible combination could code for up to 16 amino acids. Still not enough. But 3 DNA letters provide more than enough combinations to code for all 20 amino acids.
Right, so you need the sort of the code that you're going to see, and you can look in the book about this. It's always going to be.
So 3 was the answer.
3.
It was a triplet code.
Now it's kind of interesting. So it's kind of interesting from a research standpoint. As you see all those leftover ones, they're generally paired with specific. Those can often also be paired. So there's some that are sort of duplicates and so forth. But what's interesting is biochemists. Someone was asking. I don't remember who it was. But was asking about the differences of we were talking about native chemical ligation and why you wouldn't just have an organism do it and so forth. And I was talking about non-natural amino acids. Sometimes what people will do is they figured out ways to sort of use some of those excess base pairs to actually code the system to make a new non-native base pair actually be incorporated so that they can sort of use those excess to perhaps use the organism to do some of these different things. So I thought that was kind of interesting, and that ties into something we were talking about yesterday. Okay, so that's the first part, right? DNA makes messenger RNA. Messenger RNA becomes this code that effectively gives the proteins. The last step is going to be in the ribosome, so it's carried to the ribosome, and within that ribosome, the mRNA, the machinery is actually going to take place. It's going to make these proteins. And so that'll be the last video that I'll show you guys. And that's going to be translation.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the 3-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of 1 large and 1 small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape.
Those things here, these things here with the red tails, I'll just sort of tell you guys. I think it might say it, it might not, I forget. But that's going to be the tRNA. So that's going to be that's what we were saying is going to have the code. It's a small RNA sequence that at the tip of it has an amino acid. So if you notice at the end of these is that little red part, that's going to be the amino acid. And as you'll see, what's coming out of the protein is the red protein. And so basically the green part is sort of the RNA, minor oligonucleotide, and then the red part is going to be the amino acids. It might say that, it might not. I feel like it might not.
Amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon, the codon...
You guys see that? How you've got this match between the tRNA and the mRNA. And it's that sort of match that effectively aligns the appropriate amino acid sequence to actually end up in that position. So it's the match between the tRNA and the mRNA which effectively allows you to get that sort of sequence.
Each codon on the mRNA matches a corresponding anti-codon on the base of a transfer RNA molecule.
Right, so that would make sense, right? If you've got that same base pair network, right, would allow you to match, right? If you've got, say, AAA as your sequence, then if you had a transfer RNA, it would be TTT. You would get that same small hydrogen bond network, right? And then it would end up putting the, yeah. Oh, yeah, good point. Because we're an RNA. So, good point. So, but it would be UUU, in which case then you would have the sort of amino acid that would be up there. Thank you for that correction.
The subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
Quite literally. So anyways, I thought those videos were pretty cool. They give you sort of a sense for, that's kind of the interesting thing, right? Organic chemistry, we've been talking about these sort of defined things that we can maybe talk about in terms of small molecules on here. But the sort of next phase in potentially your education in chemistry, if you're inclined to do so, is potentially biochemistry. And once you get to biochemistry, you're going to be really talking about a lot of sort of bigger complexes, and you'll start seeing things like this sort of machinery that's all based upon a lot of these building blocks that we've seen, but certainly I at least think is sort of way more complicated. Okay, so we've got about 7 minutes left. I said I would potentially let you guys early, but I want to tell you guys one more thing. So that's kind of what I want you guys to know about oligonucleotides. Again, I think that's fascinating and something that you guys, it's worth it to know, and I think it's important to know in terms of your understanding of biology, especially those of you that are looking to go into medicine. Again, from an organic chemistry standpoint, types of things that I would like you to be able to do is recognize the nucleic acid if you see it. I'm not necessarily going to hold you to memorize them, but if you see, say, uracil or something like that, I expect you to recognize that that's a nucleic acid and not say, for example, a lipid or something, right? So, so that's sort of where I'm more interested in having you guys be able to recognize these structures, and hopefully you guys can do that, because that's going to be really important as we start going forward and talking about different things in a biological context. I often like to, as you guys know, I like to sort of give a spin on what we talk in terms of things that might be closer to my own research. And given the fact that we have like 7 minutes or so, I thought it would be worth it to talk about viruses a little bit. And specifically, some of the research that we've been doing geared towards HIV-based therapeutics. Okay, so viruses, and specifically retroviruses, one of the mechanisms through which they infect is based upon a lot of this nucleic acid-based chemistry as well. You'll often hear of again, this is all transcriptase, and oftentimes when you're talking about viruses like HIV, they have something called a reverse transcriptase, okay? So instead of having DNA, right, be created and then turned into RNA, they do the opposite. They'll basically have generally an RNA-DNA hybrid sometimes, but that will effectively serve as a template to generate new DNA. And what that DNA is then done is injected into the host cells, right? So viruses aren't really living, right? They require something else. They're almost just like genetic information that can be lethal, right? But they certainly spread and everything else. So something like HIV, what it is it's a set of machinery that basically creates DNA that's self-replicating, but it requires a host cell in order to infect it. Okay, so something like HIV. Right, so HIV, the reverse transcriptase of HIV is basically this giant machinery enzyme. Okay, and it's effectively got two sites. Well, it's got multiple sites, but it's got two catalytic sites. One is the DNA polymerase site. So you might have heard in the previous one them talking about RNA polymerases, right? It will have sort of a DNA polymerase site that's somewhere over here. And over here, what it will have is the RN, usually it's called the RNase H site. Okay? The RNase H site is effectively what happens in HIV, sort of generally speaking, is that the virus's reverse transcriptase will take this sort of RNA-DNA template, okay? And it will use this RNase H site to effectively cleave the RNase H to create individual subunits. That then is termed somehow transformed and then used within this DNA polymerization process to now make the new DNA. So there's an overall machinery here that's sort of essential for DNA replication. If you look in the literature, in fact, over half of the drugs, there's like 27 drugs for HIV, and like 17 of the 27 target the sort of DNA polymerase site. Okay? What they often are is you may see drugs on occasion, especially if you guys become doctors and so forth, especially if you're an antiviral doctor. You'll see a lot of drugs that are nucleic acid derivatives. So one of them that you might see, for example, is, have you guys ever heard of AZT? Right? So AZT, for example, I just want to make sure I draw the structure right. AZT is kind of fascinating because what it is basically it's a nucleic acid analog, okay, that looks like this, okay, and what it effectively serves is a inhibitor of the enzymatic function. So again, you're not going to be able to terminate the you're not going to be able to continue the polymerization process here because it doesn't have that extra phosphate, right? So it's effectively a mimic of a nucleoside, right? It's a nucleoside analog, right? But it's not going to propagate, right? Like once it gets in there, it's going to stop, right? And so basically what it does is it outcompetes a lot of the other nucleosides, shuts down and disrupts the ability of the DNA to actually be made by the virus, and then the virus eventually dies because it doesn't have the ability to do this. So it doesn't allow it to infect other cells and so forth. So you guys will be seeing a lot of molecules like this that are actually DNA polymerase inhibitors. And again, that's over half of the HIV drugs. And many, many viruses are also based upon, now that you guys have seen nucleosides and you sort of see these, now you might sort of understand why. The RNase H site is actually another very important and critical in the sort of ability of the virus to infect other cells, but there are no drugs that are available that target this. And one of the things that our lab is actually actively involved in is trying to develop potent and selective RNase H inhibitors that could be viruses. And that's really important with things like HIV because it mutates so quickly that oftentimes drugs become ineffective. So it's always good to have multiple mechanisms of action drugs. Okay, so that's it for oligonucleotides. And we'll do polymers next week, and then we'll have a couple review classes. Okay. Oh, sorry. Yeah. Sorry.
So how do you make sure that it's not connected to the alcohol?
Yeah. So that's there's right. So right. Sorry. So there are some differences.
It sounded offensive. It sounded offensive. I know.
Yeah. So
You're gonna want to. I'm telling you, she's telling me that next year. I'm not saying that. But it's so. No. I have a question for you. Why do you have to sell? After you're tested on after what? After you ask what you you asked me too? You showed us the whole paper. You showed us like 4 minutes. Are you kidding? I sat in the back that day next to some girl and kept looking at me like, shut up. And I was sitting right next to her.
yeah. Yes.
That's the question horrible. So when we have this drug,